This is an overview of an article titled Genetic Regulation of PCSK9 Plasma Levels and its Impact on Atherosclerotic Vascular Disease Phenotypes, published in the May 2018 issue of Circulation, Genomic and Precision Medicine. Elevated low-density lipoprotein, or LDL cholesterol, is a major risk factor for atherosclerotic disease. Circulating levels of LDL cholesterol are regulated, in part, by the proprotein convertase subtilisin hexin type 9, or PCSK9, protein. Inhibiting PCSK9 function has emerged as an effective new strategy for reducing plasma LDL cholesterol levels and risk of cardiovascular events. However, it hasn't been clear whether circulating levels of PCSK9 can be used as a biomarker for the presence or progression of atherosclerotic disease. Circulating levels of PCSK9 vary considerably from person to person. Some of this variability may be influenced by genetic factors. In the paper under discussion, the authors aimed to identify the genetic variants associated with circulating PCSK9 levels and to investigate potential causal effects of PCSK9 on a variety of atherosclerotic vascular disease phenotypes. First, they performed a genome-wide association study, or GWAS, of circulating PCSK9 levels in 3,290 patients with suspected or confirmed coronary artery disease. Next, they analyzed allelic heterogeneity at the PCSK9 gene locus. Lastly, they used Mendelian randomization to identify potential causal effects of PCSK9 on atherosclerosis in the coronary, carotid, and peripheral arteries. The results of the genome-wide association study for circulating PCSK9 levels in coronary artery disease patients are shown here. 31 variants, representing 5 independent signals, reached genome-wide significance. The strongest association was observed for the single nucleotide polymorphism, or SNP, designated RS11591147, which encodes a well-known missense mutation in PCSK9 that is thought to protect the PCSK9 protein from proteolysis and reduce secretion of PCSK9 into the blood. Interestingly, 4 of the 5 independent signals were located within or near the PCSK9 gene on chromosome 1p32, indicating significant allelic heterogeneity in this region. The pattern of linkage disequilibrium across the PCSK9 locus and the 4 lead SNPs from the locus are shown here. Together, these 4 SNPs explained 4.4% of the variation in circulating levels of PCSK9. 3 of the SNPs remained independently predictive of PCSK9 levels when tested in a multivariable model. Beyond the PCSK9 locus, the authors identified a genome-wide significant locus at chromosome 7p22.1 in close proximity to FBXL18, a gene whose function is largely unknown. They also identified 7 suggestive loci, just below the threshold for genome-wide significance. Importantly, the GWAS findings remained unchanged when the analysis was repeated for the subset of 2,022 patients who were not taking statins, confirming that these associations were not confounded by statin treatment. The availability of coronary angiography and other atherosclerotic disease assessments enabled the authors to test the causal impact of circulating PCSK9 levels on these phenotypes. Using a Mendelian randomization approach, with the lead SNPs near the PCSK9 gene as instrumental variables, they detected evidence for a causal relationship between PCSK9 and atherosclerosis in the coronary, carotid, and peripheral arteries. Specifically, Mendelian randomization estimates showed a causal effect of circulating PCSK9 levels on coronary artery disease status and severity, carotid plaques, carotid intima media thickness, and peripheral artery disease status. In summary, The authors identified 4 independent signals at the PCSK9 gene locus that explain 4.4% of the variation in circulating PCSK9 levels. The authors also found a novel locus on chromosome 7p22 and 7 suggestive loci associated with plasma PCSK9, although these require independent replication and identification of the causal genes. Lastly, using Mendelian randomization, they found evidence for a causal effect of PCSK9 on coronary artery disease and other vascular phenotypes. Together, these findings reinforce PCSK9 as a valid therapeutic target for atherosclerotic cardiovascular disease.